So good evening, everyone. Welcome to sixth lecture. So you guys know all about me, so I'll skip these slides. And yeah, so in the previous class, we talked about droplet-based bioprinting, right? We learned what are the different modalities, how it works, how each of these modalities work. And also we talked about what type of biomaterials are used for droplet by bioprinting, what kind of advantages are there in using one type over the other. So today's lecture, we will understand application of this technique by going over a research article. So briefly, let me remind you about all the different modalities of droplet-based bioprinting. Okay, so we learned that there were three umbrellas. There was inkjet, acoustic, and valve-based bioprinting. Under inkjet, you can have continuous inkjet bioprinting. So when we are talking about inkjet, first thing that should come to our mind that there is a nozzle at the tip of the printer cartridge or fluid holding chamber, whatever you call. And from the tip of the nozzle, drops are coming out. All right. So one disadvantage of this technique would be the shear stress that the cell-loaded droplets can experience when it is coming out from the nozzle, right? That's the one disadvantage. So in continuous one, what is happening? There is a continuous pressure being applied on the fluid chamber, because of which drops are coming out continuously in a stream. A stream of drops are coming out. So it is not very suitable for bioprinting because you're working with very expensive cells, and also the biomaterials are not that cheap as well. So this technique, if I use for bioprinting, it can become costly. All right. So it is not very much translates or translatable in the bioprinting scenario. Whereas drop on demand, where you create drop when required, all right, that reduces the wastage. So drop on demand uses different type of actuators. Actuators provide stimuli for drop to get formed. So you can use thermal actuators. We saw in thermal actuators that there is a heating coil that gets heated when you apply electric current, and it heats up to a very high temperature, like 200 or 300 degrees Celsius, at a local area in the fluid chamber, which creates a bubble like this. So when this bubble becomes large enough, it collapses and creates a pressure pulse. And that pressure pulse is enough for the bioink to overcome the surface tension at the tip of the nozzle, and thus a droplet is generated. Whereas in piezoelectric one, we use a piezoelectric actuator. So these materials, they can change their shape when electric field is applied, or they can generate electric field when it changes its shape. So both is possible. So in this scenario, we are applying an electric field that causes the piezoelectric actuator to change its shape and deform the fluid chamber, right? It will cause radial deformation that creates a pressure pulse, and thus a droplet is generated. Whereas in electrostatic one, here we use again electric field to apply some charge, all right, on a pressure plate. So this pressure plate in presence of electric field can change its position, all right, and in absence of electric field, it comes back to its original position. So due to this change in position of the pressure plate, a instantaneous change in the volume of the fluid chamber gets changed, and that causes again a pressure pulse to generate and a droplet gets formed. And another technique that comes under inkjet bioprinting is electrohydrodynamic jet bioprinting. Here, what's happening? Here we apply electric field. One end of the electric field, that is, let's say positive end, is connected to the tip of the nozzle. Another is connected to, let's say, substrate. And as you apply air pressure on the fluid chamber, bioink fluid chamber, the ink comes out in a steady flow rate. But as soon as it experiences the electric field outside it, it it breaks up into droplets. And these droplets are basically charged at the tip of the nozzle, and this charge is enough to overcome the surface tension, and because of which droplets are formed. And these charged droplets, these are positively charged, let's say, because there is positive end at the tip of the nozzle, it is attracted towards the negatively charged substrate. In this way, you are controlling the deposition of the droplet by using charge, and that charge you're applying through electric field, right? And if we are not interested in using inkjet bed bioprinting because of the main disadvantage that it can cause shear stress to your cells, then there are other two methods that does not use a nozzle, very does not use a very tiny diameter nozzle, because in these techniques your diameter of the droplet is not dependent on the nozzle size, whereas in the inject ones you can have options of using various nozzle sizes so that you can modulate your diameter, nozzle diameter, droplet diameter. First one is the acoustic bioprinting. Here we use sound wave, all right, to generate droplet. So what happens? There is a piezoelectric material, and to it there is gold rings attached, interdigited gold rings attached. So when you apply electric field, the actuator changes its shape, and that basically also causes generation of sound wave through the gold rings. Now this sound wave, it travels through the bioink reservoir, which is just below it. And as the sound waves are formed, they are usually circular in structure. Like the sound waves are circular, and the center of the circular sound wave is where the pressure is highest. All right. And that is also where the outlet is there for the bioink to come out. So this sound wave in this way creates a pressure pulse at its center, and a droplet is ejected out, all right, from the tip, from the end of the reservoir. Okay. Whereas so here the advantage is that you do not have to use a nozzle or not, needle, right? But the disadvantage could be that you cannot use viscous materials here. You need very low viscous material so that the sound wave can propagate well. And also the cells, they are not exposed to any high temperature or physical stress, like due to piezoelectric actuators you apply some stress, right? So those stuff does not happen. The cells are completely free of any kind of stress here, neither electric field you need, like nothing. It's a very gentle technique. In microvalve bioprinting, here what happens, you use a microvalve connected with a plunger, and this plunger basically gates the tip of the fluid chamber. So the tip of the fluid chamber is basically conical, but it is not, but it is having a big diameter. All right. The diameter of the aperture is quite big. It is not very small as we saw in inkjet-based bioprinting. So shear stress is not the big problem here, all right? The big problem here could be there is not much resolution of droplets. The resolution of the droplets that you get is very high. You don't get, sorry, is very slow. The resolution of the droplet that you get is very low because you get very big size droplets. All right. You cannot go very small size droplets here. But here we are controlling the ejection of the droplets by basically controlling the movement of the plunger, and this movement of the plunger happens through a microvalve system. This microvalve system gets activated in presence of electric field, and when electric field is applied, the plunger moves up. It allows the bioink to come out. When the electric field is switched off, the plunger moves down. The bioink cannot dispense out, right? So in this way, you're controlling the ejection of droplets by basically controlling the movement of the plunger, that is through a microvalve system, right? So these are the few modalities of droplet-based bioprinting and a brief description of how they work. Okay, so in today's class, as promised, we will talk about a research article on this topic. So this is the article that we will discuss. It was published in 2017 and in the Journal of Scientific Reports. It's a very good article in my opinion. Um, I couldn't find a very recent article on droplet-based bioprinting, like maybe published in 1 2 years. It's because nowadays people are moving more and more towards extrusion-based bioprinting and DLP. DLP stands for digital light processing bioprinting. Droplet-based bioprinting is not at a very high stage right now. Okay. But yeah, at that time when the bioprinting started, it started with droplet-based bioprinting, which I already explained. So before I dive into this paper, if you have any doubts related to last lecture that we discussed, and if you have any concerns or any comments to share, please, now is the time.
Ma'am, whatever 3D printing times we have studied so far, the
Mhm.
mechanism is only the only difference, right?
Yeah,
Or is there any
yeah.
other difference?
So the way the technique works, it makes all the difference. You will choose your biomaterial differently. Okay, like for extrusion one, you cannot use a very low viscous material, whereas in bio droplet based one you can. Okay, because if you use a very high viscous material in droplet based one, you cannot get nice droplets. So the mechanism of how the technique works makes all the difference in your choice of biomaterial, in how much cell density you can use. Also, resolution will differ, all right? Like how big or small features that you can bioprint. Uh, but all of these techniques have the possibility to create similar tissue types. Like you can make skin tissue with both extrusion one and with droplet one. People have demonstrated that. So the tissue types, they can be like same tissue type can be prepared with different techniques. Only thing is the how well you are preparing. So it's possible that in droplet based one you can be limited by few cell types, whereas extrusion based one, the systems usually come with multiple print heads. I have seen a machine with six print heads actually. So you can print 6 cells at a time in extrusion based one, whereas droplet based one, the systems usually come with one print head. So you have to like shuffle between bio inks, like one bio ink after the other and after the other to print multiple cell types. So depending on what technique you use, it changes your entire process of bioprinting altogether. Okay, starting from bioink preparation, as we discussed, right, to processing, post processing. But all these bioprinters will use the CAD file, STL file that we discussed in the very beginning. So that thing remains the same. And also another thing that remains the same is the tissue type. You can make same tissue type with different techniques. All right.
Ma'am, you said in extrusion we cannot use low viscous, right?
Yeah, it will not
But
extrude well. Yeah.
uh, is it right, ma'am?
Yeah, it's right. You're right. Yeah.
But you said at the same time people are moving towards extrusion, not using droplet or other methods. But we can use a low viscous. Can we still go with extrusion?
Can you come again? Can you repeat your question again?
Okay. If you want to use a low viscous bioink, is it possible
Mhm.
we can go with extrusion itself, or we need to change the method?
So if you like your application has to use a low viscous material, let's say has to use, then you have to use extrusion only. Like there's no other machine that you have only the extrusion one. Then you have to modify your bioink a bit to add viscosity. You have to add some modifiers, right, that increases the viscosity of the bioink. Then you can maybe low percentage you can add to your original material, but that will enable better viscosity, and then you can extrude better resolution filaments.
Okay, ma'am.
Okay, there is no other questions, then I'll move on. But if you have any questions in between, feel free to stop me and yeah, ask. This is the paper is titled High Resolution Patterned Cellular Constructs by Droplet-Based 3D Printing. Okay, so let's quickly go over the abstract of the paper first. That gives a general overview of what work has been described, what the authors did. So droplet or bioprinting, in fact, is an emerging technique for the fabrication of living tissues that allows cells to be arranged in predetermined three-dimensional architectures. Yes, we know. However, to date, there are limited examples of bioprinted constructs containing multiple cell types patterned at high resolution. So since this paper is 2017, they are talking in that context. But now we have multiple papers, all right, multiple research showing that we can pattern large number of cells, like three, four, five cells. I've seen papers, people are doing it, all right, and at good enough resolutions too. But here the resolution that they're talking about is really good. Okay, this is the first paper where I saw such high resolution patterning of cells. Okay, so here we present a low cost process. So the process that they are proposing is low cost, all right, because the materials that they are using at a very low volume. Droplet based bioprinting allows you to use less volume of material so that you can conserve on material cost and the cell cost as well. So this is a low cost process that employs 3D printing of aqueous droplets containing mammalian cells to produce robust patterned constructs in oil. Okay, so they're making a pattern construct in oil. Remember that. All right, we will look into it later in more detail. So which were reproducibly transferred to culture medium. So they printed the constructs in oil and then they transferred to culture media for post processing. They used Haig cells, which is the human embryonic kidney cells. So these are immortalized cell line that they used in this paper. And also they used ovine mesenchymal stem cells. So they used two types of cells. Basically, they showed the use of two types of cells with their technique to show the robustness of their technique. And they were printed at relatively high densities. So if I guess both of you were present in my tissue engineering course, which I took. last year. So there we talked about clinically meaningful cell numbers, right? So our tissues, or you can say our body has around 10 to the power 14 million cells, and if you talk about at the tissue level, it is around 10 to the power 5 to 10 to the power 7 cells. So you need that much number of cells to make a tissue, because that is the cell number that you find in your native tissue. So the cell number that they have used, it as is at the same level of the cell number that is already found in our native tissue. So remember this as well. This is an important point, that they used a very high cell number, that is 10 to the power 7 cells per ml. And the droplet resolution is also quite high. So they patterned using droplets of size 1 nanoliter. And using 1 nanoliter droplets, they made a 3D structure. The 3D geometries that they were printing had features which were less than 200 micron, means the features were very small. Okay, you can only observe them through microscope. And they made various features including cell junctions, diagonal plane junction, and also osteochondral interface. I'll show you. Okay, the printed cells showed very high viability, which is close to 90% on average. That's good. Hex cells within the printed structures were shown proliferate under culture conditions. And significantly, a 5-week tissue engineering study demonstrated that O OMSCs, that is ovine mesenchymal stem cells, they could be differentiated down the chondrogenic lineage to generate cartilage-like structures containing type 2 collagen. So they printed mesenchymal stem cells, then added cues, growth factor cues, to cause differentiation of these mesenchymal stem cells into chondrogenic cells, chondrocytes basically. And they proved that these chondrocytes were showing the marker, all right, specific biomarker, as well as it secreted collagen type 2. Okay, so their technique, to show the power of their technique, they used in situ, basically, like after printing the mesenchymal stem cells, they caused differentiation of the cells. All right, cells were still in the 3D structure. So that's a cool thing that they did. So now the key points are well highlighted in your mind. Now we will go over the work one at a time, with one slide at a time. So first, let's discuss why did they use the printing in oil, basically. Why did they made 3D constructs in oil? How is their technique would be better than the existing state of the art that is there? So, extrusion-based bioprinting, they say that this technique is ideally suited if you want to make large constructs, which are much bigger in volume, like greater than 1 centimeter cube. Makes sense, right? You're using filaments, which the size of filament is obviously bigger than droplets. So if you're using filaments, the size of the construct would be quite big. In fact, in my experience, you can get good resolution structures using extrusion-based bioprinting. Um, if you don't go below 10, 10 mm by 10 mm, okay? If you go below, let's say I'm making a grid like this, the length and breadth is 10 mm. If I go below than 10 mm, all right, then it's very hard to get good resolution. The features does not become prominent. So um, usually that's why I usually start by testing structures which are of at least 10 mm by 10 mm, okay, or higher. Even and like if you go by 20 mm by 20 mm structures, those are those have the better resolution, obviously better resolution than 10 mm by 10 mm one. Okay, so that's why it's said here that extrusion based bioprinting is ideally suited to make large structures. And but one disadvantage is that it cannot, or it poorly patterns multiple cell types in high resolution. So it's possible to pattern cells by controlling the layer type. So let's say you pattern cell in layer 1 to 3, okay? And then you can be sure that your three layers have one cell type, and then you pattern the next cell type in subsequent layers. So in this way, you can pattern cells very nicely. Another way to pattern is you can make crisscross patterns. Let's say these vertical lines correspond to one cell type, and the horizontal line, it can correspond to another cell type. In this way, you can pattern two cells in one layer itself. Or similar type of infill by this is called infill. This design that I drew inside is called infill pattern. So in this way, you can control the infill pattern, all right? And using the infill pattern, you can pattern the cells. So if you want, you can print three types of cells in one layer. Like, let's say vertical line is one cell type, horizontal line is another cell type, and boundary, that is periphery, is another cell type, right? But you cannot go any lower dimension. Means you cannot pattern like this with good reproducibility. Like, let's say this is a filament, right? Let's say this is a horizontal filament, and you want to pattern only this section with one cell type, okay, and then leave a gap, let's say, and then another section with the same cell type. So in this way, you cannot pattern very well. It will become quite challenging to do it, okay. um, that's why it says that it has a poor ability to pattern multiple cells in high resolution. Okay, you can do this. I'm not saying it's impossible to do it, but but you will face more problems in doing this. Whereas this approach, which I explained just now, is much easier to do it. You can control the G code uh, in a better way. Here, controlling the G code means controlling the movement of the XYZ axis to make sure that you get exactly this type of pattern. It is bit challenging. It is bit more challenging. Okay, so yeah, um, these are the advantage and disadvantage of extrusion one according to them. And there is another technique that we haven't discussed yet. We will start discussing this in next week. We will discuss it next week, and after this we have one more. So in this course, we will talk about four bioprinting techniques. This is the third one. So the laser-based bioprinting, I briefly discussed it during our introduction. Here, what we do, here we use laser pulse to deposit droplets of bioink. Okay, and um, how it is done, there is a layer of bioink, and on top of which there is an absorbing layer. The laser falls on the absorbing layer so that the absorbing layer extracts the excess energy of the laser pulse, and wherever the laser is moving over the absorbing layer, the bottommost, the droplet layer, also experience that pressure. Okay, and that pressure pulse is enough for ejection of droplet. In this way it happens, but we will discuss in more detail in next week. Okay, so this technique advantage is obviously it avoids extrusion. When we talk about avoiding extrusion, that means we are avoiding any shear forces that can cause cell death. But disadvantage is it's a very high cost technique because of the cost of the laser is quite high, and it is not very well designed to make 3D constructs. It has been used mainly to pattern cells in 2D, but To make 3D structures, it is bit more difficult, all right? It means I'm not saying that it hasn't been attempted, it has been attempted, but it's slightly more difficult. Okay. And lastly, if we use droplet based printing, let's say inkjet or valve based one, here you can face issues like in inkjet, you can face issues of clogging. There are limited type of power materials that you can use that matches the requirement of the technique. And also the cell density that you can use is quite suboptimal. Okay, you cannot use very high cell density because again it can cause the issue of clogging. And also due to extrusion process itself, as it uses a nozzle, it can cause cell death even if you are loading very high cell numbers. Okay, but as valve based system, its inherent issue is that it cannot print droplets of very small size. Okay, it makes droplets of big sizes, so it allows low resolution bioprinting. So these are um, the points that compares their technique with the existing one. Okay, now we will see why did they mention these points in the paper. Okay, and how their technique is able to address these points. So
Ma'am, am I audible?
yes, you're audible. Go ahead.
Ma'am, you told that this extrusion is nowadays widely used. So
Yeah.
this property that um, like the cells are damaging by the shear stress. So how we are helping or how we are overcoming that drawback of this extrusion based bioprinting.
Okay, that's a good question. So, in order to overcome the issue of shear stress, you have to play around with the parameters of the technique itself. The technique comes with a lot of parameters, like I mentioned previously. You have to worry about the bioink properties, the biomaterial properties, in fact, the rheological properties, its viscosity, its shear thinning capability, its like strength, physical stiffness, and all. You have to worry about that. Then you can also play around with the printer parameters like uh, f- flow rate, volumetric flow rate, then extrusion pressure, the speed at which the printhead is moving, the temperature, and then light properties and everything. So when you do a lot of permutation combination type of study, like keep one parameter constant and other variable, when you do that, you can see improvement in your cell viability. Okay, so we prefer to use shear thinning biomaterials because if you don't use a shear thinning one, then cells are much more subjected to shear stresses. But shear thinning materials itself has the property to, reduce its viscosity. So if the viscosity is reduced under shear, then the cells can flow better, right? It can flow better through the tip of the nozzle, and the stresses that it will experience is much less. So that is one way, all right? But yes, you cannot fully avoid shear stress in this technique. Another reason that it is becoming more popular is that we have more number of shear thinning biomaterials available with us. So there are more open-ended research questions now, like how these biomaterials can be used for extrusion-based bioprinting. And also, if you see logically, for the researchers who are just starting to learn bioprinting, it's much easier and handier for them to learn extrusion because we have been extruding throughout our life. Like when you um like pull when you use a toothpaste tube, right? You're extruding basically. Or when you use a honey bottle, right? Or when you use a ketchup bottle. So it is much easier for researchers to understand extrusion, the mechanism of extrusion. The systems are very easily made. Like you see, there is a cartridge with the tip is a nozzle or a needle, all right, and there is some pressure mechanism, like air pressure is there, or some screw or piston is there. So, and also like, so if you think in these points, that is why extrusion is becoming more and more popular, even in people who have no prior experience with additive manufacturing. All right. So these are some reasons why it is becoming more popular. Whereas if you see in droplet based one, the technique that the introduction technique that we saw yesterday, so not yesterday, last week, we saw that only limited materials fit into the um, requirements of the technique, right? There was mostly people were using alginate for making droplets because it has low viscosity. Whereas in nature, if you see, there are more shear thinning materials. So these are some factors, and we hope that it will change soon with the invention of or discovery of new biomaterials, all right, and development of new types of bioinks too. So yeah, because of those reasons, extrusion is much becoming much more popular, I feel. Did I answer your question? All right. I hope you feel better. Is it too cold there? Where are you right now? I heard that your voice is not good right now. Maybe some throat infection.
Yes, ma'am.
Yeah. I recently, my mother also having similar throat infection. She is also not able to speak. I'm yet to experience India's winter right now, but in Houston too, believe me, it's cold here. I expected that in Houston it won't be, but yeah, it's cold, but it's not as cold like I'm used to in Kanpur or in Delhi. It is not as cold as there. So yeah, I hope you feel better. Yeah. Moving on, let's discuss their work. How did they 3D print their constructs? Let's understand their process. I'll quickly go over the points, all right, first, and then with each point, I'll show you the images and everything. If you have any queries in between, let me know. So the 3D printer that they used is actually a droplet one, all right, droplet based 3D printer, and the droplets are formed by drop on demand using a piezoelectric actuator. Now the printer head is static here. That also you must note. The printer head is static. It cannot move in XYZ direction, and it has a piezoelectric actuator to generate droplets. So it's a drop on demand printer with piezoelectric actuators. And the placement of the printhead is such that the the tip of the nozzle is completely immersed in an oil bath, lipid oil bath. I'll explain why did they use lipid molecules. So they used oil bath with lipids in it, and the nozzle was completely immersed. And why they immersed it? So that whatever droplets that gets ejected, it will fall directly into the oil. Okay. And here, which component is moving? It is the stage that can move in all XYZ directions. Okay. Stage can move, and through the movement of the stage, we are controlling the position where the droplet will fall. Okay. What's the bioing that they used? So they used agarose. They used ultra low gelling temperature agarose, and this agarose was mixed with FMOC dipeptide solution. So these are basically dipeptides, all right, dipeptides. They can be diphenylalanine or isoleucine glycine combinations, and these peptides are protected by a um, chemical group that is short formed as FMOC, but it can be expanded into fluorine fluoronyl methyloxycarbonyl. All right. So I've written the full form here at the bottom of the slide. Okay. So why did they use these dipeptides? Because they can bond with each other, but the weak bonds they can form with each other and form a weak hydrogen network. Okay. So with agarose, they mix this FMOC dipeptide solution, and also they mixed serum free cell culture media, and with so cell culture media is added to sustain the um, viability of your cells. And they made two types of bioinks, one without collagen and one with collagen. Okay, this is the bioink that they used. What's the cell density did they use? So they suspended around 1.7 into 10 raised to the power 7, sorry, 1.5 into 10 raised to the power 7 cells per ml. So in the order of 10 raised to the power 7 cells, all right, in a very small volume of bioink, that is 100 to 200 microliter, right? So the cell density is quite high in the overall bioink solution. What's the composition of the oil bath did they use? So they used uh, 35 is to 65 volume by volume mixture of silicone oil, sorry, a mixture of undecayed to silicone oil AR20. Okay, and this oil mixture had some lipid molecules inside. So what is the name of the lipid molecule? It is 1,2-diphenyl tanolyl SN glycerol 3-phosphocholine is short formed as DPHPC. Okay, so this is the lipid molecule, and the oil is basically containing undecane and silicone oil. All right. So this is the entire setup. They used an oil bath. They used a drop-on-demand bioprinter, droplet-based bioprinter, and the droplets are ejected through due to piezoelectric actuators. The nozzle is completely immersed into the lipid in oil bath. The stage can move. Stage has the bath. So basically, the bath can move in XYZ direction. The bioink contained serum-free cell culture media with agarose. Okay, the agarose type is ultra-low gelling temperature agarose, means it can gel at a very low temperature. With this agarose, they also mix some weak gelators, weakly bonded gelators, that is FMOC dipeptide solution. And they use the bioink with or without collagen, depending on the use, all right, depending on the application. And they added a very high density cell. They added very high cell density. So that is around 1.5 into 10 to the power 7. Okay. So I'll show you one image quickly to understand what I just said. So this is the stage. All right. That stage can move, onto which there is a glass surface, and on the glass surface you have the oil bath. So this yellow background have the oil. Okay. Silicone and undecayed mixture. And into this you can see these tiny particles. This one, these are lipid particles. All right, you saw one lipid that they added. And this is the nozzle. This nozzle is completely immersed into the oil bath. This nozzle, you see, have the bio cell laden bioink, and as the actuator is working, it is ejecting one droplet. But there is another interesting thing that is happening. The as soon as the droplet is coming out into the oil bath, a monolayer of lipid gets formed around it, because you see there are free lipid molecules. They come and interact with the droplet and forms a monolayer. And as they are positioned in anywhere in the XYZ direction, this lipid monolayer forms a lipid interface with the other droplets like this. You can see these droplets are basically bonded with each other through lipid interface. And in this way, they have positioned it. All right. So, yeah, this is the actual image of how the printing is happening. This is the nozzle again. These are the droplets, all right. And with the ejection of one droplet, that are surrounded by lipid molecules, okay, they are being positioned in a 3D structure, right? So this entire construct is in oil, right? Cool. So quickly, I'll go over. So the why did they use go over the points? So why did they use oil mixture? First reason that comes to our mind is that if you do it in air, if you eject droplets in air, then you will face the problem of splashing and spreading, right? If you and you can solve that problem by basically making sure the velocity of ejection is not too high or not too low. We discussed this in the last class, right? But still, even if you eject the droplets in air and you solve the problem of splashing and spreading, you cannot make sure that the droplets are fully spherical. There is a the droplets will become like this on a surface. It will become like this, right? When you position it in air on a stage. So the resolution of the 3D structure that you can make basically lowers down. But here, since they are using oil, they are controlling the speed at which the droplets are descending first, because the gravity uh, will work slowly here because oil is much more denser, right? And also there is one more advantage of using oil is that you can make sure the droplets retain their size, retain their shape, sorry, they retain their spherical shape, all right? And as they are getting deposited, you will see that they are forming an hexagonal array, sorry, they're forming an hexagonal array. I'll show you in the image. Oil also helps in making sure that your drops don't get freezed, all right? They don't experience a very high cold shock when they are going to form gel at a very low temperature. So the agarose that they're using, I told you it gels at a very low temperature. So I'll show you later that what they are doing after printing in oil, they are gelling the entire construct, because in oil it is all drops. And to preserve the shape, they have to keep it at 4 degrees Celsius for a few minutes, like 20-30 minutes they have to keep it. So during that period of incubation at low temperature, oil preserves the structure as well as prevents freezing of the droplets. Okay, and the technique that the printer that they're using is able to print very small drops, like 1 nanoliter size droplets into the oil. As the droplet is coming down from the tip of the nozzle into the oil, the lipid molecules, they form a monolayer around it, and as they're positioned in layers, okay, they form a droplet interface bilayers. Like these monolayers, they um, interact with a monolayer of other droplets and form a bilayer, like we see in cell membrane, right? So this bilayer formation preserves the structure, all right, as more and more droplets is getting formed. So you know a lipid has a hydrophilic and a hydrophobic end, right? So depending on the environment, if the environment is hydrophilic, then it can change its orientation and expose its hydrophilic side. So in this way, the lipid orientation can change. So if this is a lipid molecule, so and the circular part is the hydrophilic portion, and the tails are hydrophobic, then the hydrophobic tails will tend to stay towards inside. So similarly, if this is a droplet with tails of hydrophilic coming out, hydrophobic coming out, and the hydrophilic end stays inside. Obviously, oil is hydrophobic, so you will have droplets that has a lipid monolayer like this. So as more and more droplets are getting placed, in a layer-wise fashion. The hydrophobic ends, they will interact with each other, right? And they form a bilayer. What they form? They form a bilayer. So a transient, a quick, basically, quick formation of bilayers is there in between the adjacent droplets, and that holds the entire structure. And they are packed into hexagonal array. So they imaged the packing. They imaged a 3D structure of oil droplets. Okay, they put some yellow colored dye to help us visualize the hexagonal shape. So this is the boundary of a droplet, and as they are getting deposited one over the other, the spherical shape is lost, rather a hexagonal shape is attained. All right, so in this way they are saying that the packing of the droplets happens in order to form a hexagonal array like this, and this is one layer over the other. All right, and here for imaging this, there is no cells inside. It's just a cell-free bio ink, all right, for this. And the technique is quite fast, means they can make 1 mm cross 1 mm cross 0.5 mm construct very quickly. That is, in 5 minutes they can make it. But they have also attempted to make larger structures as well. Okay, so this technique overall helps us realize that they can print very high resolution droplets basically with a large number of cells without losing the shape of the droplet. And how they are retaining the shape? They are retaining the shape by basically using the oil bath, then also using the dipeptides and agarose, right? And also they have shown in the supplementary that if they omit the FBS, that is the serum, fetal bovine serum, if they omit it from the bioink composition, that also helps in retaining the shape of the droplets in the 3D structure. Okay, so in this image, you see E and F. This is an image of the cuboidal shaped 3D structure that they made out of droplet. Here, they have stained the live and dead cells with two different dyes. Calcine AM stains live cells into green, whereas propadium iodide or PI stains the dead cells red. So this is this imaging that they have done immediately after printing. So they have not transferred it to cell culture media. Okay, the printed construct was in oil, then they added these two dye and then they imaged it. This is in low resolution. When they increase the resolution, they are showing the boundary of the droplets. You see, this is the boundary of the droplet, right? And inside the droplets, there are large number of cells. Okay, so this is the image of how the setup looks like, kind of. So they are using a cuvette, a glass cuvette that is positioned over the motorized stage. And into this glass cuvette, you can see a big droplet-based structure is formed. All right, so this is a 3 mm by 3 mm structure. So typically they are working with 1 mm by 1 mm structure, but their technique can also print larger structure like 9 times the volume, right? So this is the image of the large structure, and this is the cells, how the live and dead cells are positioned within the large structure. Now, another thing to note here is the cell density, how much cells are actually there in the printed structure. Okay, how much cells you can get in the printed structure using their technique. So this is the graph. So they're saying if they use a bioink with cell density 15 into 10 to the power 6, that is basically 1.5 to 10 to the power 7, then the resulting cell density that they can get in the printed structure is around 3 into 10 raised to the power 7 or 13 to 10 to the power 6 cells per ml. All right. So the cell density is quite high in the printed structure. They basically calculated it by manually counting the cells within the structure. They are saying that there are around 38, 1 to 38 cells per droplet. Okay. And depending on the volume of the structure, they have come to this number. And they attribute this, why this is happening, because the cells, they can sediment at the printer nozzle. So as the bio ink is loaded, since cells, they are of small size, the gravity will pull them much faster, pull them down much faster. So you can see from this image, or you can visualize from this image, that cells will have a more tendency to come and settle down at the tip of the nozzle. Okay, so that attributed to a very large number of cell per droplet, and ultimately large number of cells per printed construct. Okay, so they say, since this is the result, since they are getting very high cell density printed construct with a very small volume of bio ink, uh, so their technique is quite appealing if you want to fabricate 3D structures of tissues with very high value cells. All right. If we are using cells whose extraction is quite expensive, you need more resources to do that, or it's very difficult to get. If you're working with these kind of cells, then their technique works very well. Then they did live dead assay, like I showed you in the image. And from the image, they are saying that the cell viability is close to 88%. All right. And similar type of cell viability they are reporting for non-printed cells. Okay. If you just encapsulate the cells in block of the bioink, like without subjecting it to any kind of printing mechanism, or when you just simply eject out the cells with droplets, like cells loaded inside the droplets in air. So these are the two control experiments that they did to compare their method. And what they're saying is that the control methods also have the same viability, cell viability, as their method. So neither the bioink that they're using, neither the piezoelectric stress is causing any cell death. All right. What could be the cause of the cell death that they're observing? Means it is not yet 95, right? It is close to 88%. So what could be the cause in drop in cell number? It could be because the cells have spent a large amount of time away from the ideal conditions. Means you need some time, right, to prepare the cells, bioink and all. So it may take like 2 to 4 hours in total. And also when you do imaging, you have to keep the live construct outside as well. So those, because of those things, they say that there is a drop in cell number. But when they keep it back in the ideal conditions, like 37 degrees Celsius, pH of 7.4, when they maintain those ideal conditions, the cell numbers, they boost up, means the cell viability improve. Okay. All right. Any questions so far in understanding how the printing, droplet printing in oil is happening here? Any questions so far? I'll explain a bit more on it in the later slides, but any questions on whatever I have discussed?
No, ma'am.
Okay. Good. Okay. Now, they have demonstrated the power of their technique by printing multiple type of cells, basically two types of cells in different pattern. All right. So they used HEC, as I told you, human embryonic kidney cells. The entire name of the cell line is HEC 293. They used these HEC cells and they stained it with two different dyes of two different color. One is deep red, another is red. But for representation purposes, they false colored it in the software. So one cell type will appear blue, another cell type will appear yellow. Although they are same cells, only thing is they are colored differently. Okay. So first image shows that they have made a Y-shaped construct in a square within a square construct. So you can see the blue colored heck is forming a very nice Y shape within a block of square construct. Okay. Here they formed a cross, a crucifer again. Okay. In similar way. Then they tried to make a 3D model like this, where the two populations of Heck, 1 and 2, they are separated by this diagonal plane. All right. And when they imaged it in confocal microscopy and observed the sections of the construct, all right, observe the different sections of the construct, you can see the interface very nicely. So this is at the lower one, Z is equals to 45 micron. Let's say here in the lower one, here the blue part is much more prominent than the yellow one. So there is a very nice boundary between the two cell population. And as it is going up, let's say here, okay, 192 micron, here the yellow population is much more. And this is the blue interface separating the blue cell line. Okay. Similarly, they also printed laminar constructs, means one layer over the other. So the lower layer has like three droplet thick, and the upper layer has like four droplets thick. So in this way, they demonstrated that they can make layered structures. Then some structures with interdigited interface, means finger-like interface they have made, and each finger is like one droplet resolution. Okay. So this is the image of that they obtained at z is equals to 45 micron section. Okay. So this is the interface. It's not a straight line, rather something like this. Then this is now in this image, I'm showing that they have printed two different cell types. So up till now, wherever you are seeing yellow and blue colored images, there they're using one cell type, but one cell type distributed in two different populations. One population is stained with one dye, that is, say, let's say deep red dye. Another population is stained with red dye. Okay, and they are falsely colored for better representation. Okay, whereas here they're using two different cell types, osteoblast and chondrocyte. Okay, they are again stained with two different colors, deep red and red, and they're falsely colored for a better representation using pink and yellow, purple and yellow color. So they made a laminar structured layer structure like this. The bottommost layer is the chondrocyte one, all right? The bottommost is the chondrocyte one, and the top layer is for the osteoblast. So in this way, they are showing that their technique can produce a high resolution patterns, okay, using droplets containing two different cell types, okay, like osteoblast or chondrocytes, or same cell type but stained differently, all right, so that we can visualize it better. So basically, two cell types. Using two cell types, you can make a very good high resolution pattern. All right. Now let me quickly explain you how did they go from printing in oil. All right, they printed the construct in oil, and then they transferred it to cell culture media for post processing. How did the entire step go? So first thing that they did obviously is the printing in the oil. This is the first step. And then they allow gelation. Okay, how did they do it? They basically kept the construct in the oil bath at a very low temperature, 4 degrees Celsius, at for 20-25 minutes. They kept the entire construct at a very low temperature so that the agarose that is there in the bioink, that will get gelled, that will form gel. Okay, so it will now increase the rigidity of the construct. After that is done, they had to remove most of the lipid molecules from the droplet based construct. How did they do that? They washed the construct multiple times in um, silicone oil. Okay, they wash the construct multiple time silicone oil until and unless the lipid concentration is very much diluted, like around 1% of its original concentration remained. Until that, they washed the construct multiple times with silicone oil at room temperature. Now once it is almost free of lipid molecules, they what they did, they coated the construct with a droplet of agarose only. So what they took a droplet of bioink containing agarose but without cell. Okay, this droplet is a very small droplet, like 2 to 0.2 to zero point 0.4 microliter. Okay, it's a very small droplet. They added over the bioink, they added over the printed construct. Okay, so that it gives a uniform coating to the construct. And again, they kept it at a very low temperature so that the coating becomes hard. So this coating, what's the purpose of this coating? Is to basically hold the structure much more rigidity, give a outer layer of protection. Okay, so that when you do the transferring to culture media, there is less chance that it will crumble up or it will break, because basically these are all droplets, right? You need to make sure these droplets don't fall apart, because now you have removed the lipid bilayer, and um, you need some more force to keep the droplets together. So now it is gelled. Now they will transfer it to cell culture media. So they don't just take it and drop it in cell culture media. Okay, they don't do that because it's very small construct. If you do that, the splashing one, okay, it can cause breaking of this of the construct. So that is why they use a two-phase column. So up topmost phase is oil, the lower phase is the culture media. Okay, and they take the pipette tip, okay, to like pick up the construct very gently and keep it, basically keep it on the oil phase of the column and allow the gravity to do its work. Okay, the construct will smoothly pass through the oil. All right, and uh, mm, basically pass the interface of oil and culture media and gently get placed on the surface of the well, right? So when that happens, they just remove the topmost oil layer, okay, and added more culture media. So in this way, they transferred the construct from the oil bath to culture media. Any questions? So what they observe is that this method of transferring is good, it works, because it didn't cause any damage to the dimension of the printed construct. The dimensions of the printed construct remained unchanged, and whatever cell layers that they added in the construct, the discreteness of the cell layers also remained as it is. Okay, they didn't mix up, the droplets didn't get mixed up with each other. Okay, Any questions? Any questions? Oh, okay.
No,
No
ma'am.
questions? Okay. Yeah. Uh, Now they assessed the like properties of the cells after they have transferred it to cell culture media. So first they checked the cell viability by using live assay, like I explained previously. The green cells are live cells stained with calcium AM. The red cells are stained with PI or propydium iodide. And what we see that the cell viability is like around 80%, which is almost the same as we saw previously when the construct was in oil. Okay, so there is not much difference between the cell viabilities in the context of cell viability between two conditions. And what they also say is, since dye, the live dead staining dye was able to penetrate the droplet and enter the cells, it means that the lipid bilayer has been successfully washed off, right? It means it has been successfully disrupted, and that is why they could enter. Okay. Then they are using immunocytochemical staining. Basically, they used antibodies specific for a specific biomarker. They used this type of antibody specific for a protein called phosphohistone H3. So, and what they observe is that this marker is a marker for G to M transition of the cell cycle. Okay. And see from this image at day 7 of culture in cell culture media, they observed that most of the cells, around 2 to 8% of the construct, have like white colored dots. So these white colored dots represent the presence of this phosphohistone H3 marker. All right. This means that cells are in dividing phase, right? It's in mitotic phase. They are proliferating, all right, and increasing in number. It shows that. Means their technique didn't affect the proliferation capability of the cells, which is encapsulated in droplets and then formed into a printed construct. They are dividing. They're always in dividing mode, at least at day 7. Okay, this is the graph that shows the cell viability. All right. So they checked for five constructs, and each of the five among five all have a similar viability of 80%, close to 80%. Okay. So what we understand is that the transfer to culture medium didn't affect the cell viability. It didn't affect the cell's ability to proliferate, because you see the biomarker for phosphohistone 3, that is a biomarker for the G to M transition of cell cycle, is present in 2 to 8% of the cells. Okay, so that shows that the cells are in dividing stage. So that's good. All right, so their technique works well. It doesn't hamper the basic function of the cell, that is to live and proliferate. Then to show the much more advanced, like how this technique can be used, they also printed stem cells, the ovine mesenchymal stem cells they printed, and showed that these printed stem cells can be differentiated into chondrogenic lineage, all right, and you can make cartilage like tissue, all right, in vitro in lab. So the viability of these stem cells is close to 80 to 90%, like we saw previously. Okay, there is not much change in the viability. And to trigger the differentiation of these mesenchymal stem cells into chondrogenic lineage, they used a growth factor called TGF beta 3, transforming growth factor beta 3. Okay, they added this growth factor into the culture medium. All right. And what they saw at day three, this is day three, Mitch. What they saw is uh, one early chondrogenic biomarker, SOX9, is being expressed, all right, in the droplets. You can see there it is colored as orange, whereas the blue color is representing the nucleus of the cell. So this image shows the proof that yes, the cells are getting differentiated with into cartilage cells, chondrocytes. Also at day 35, when they stained the droplets, stained the droplet containing constructs using peroxidase staining, what they showed that type 2 collagen was secreted, all right, which is a key component of hyaline cartilage. This is a type of cartilage where type 2 collagen is mainly found. Okay, so these two basically images prove that the cartilage is getting formed. Then at the molecular level, they also prove that there is expression of SOC9. So they did PCR, digital PCR, and on constructs, printed constructs, which are grown in presence of TGF beta 3 and in absence of TGF beta 3, as well as they compared the result with the standard gold standard for this experiment, that is a pelleted culture, means the cells, only cells are there. There is no droplets, only cells. They are packed very tightly with each other into a pellet. So that's the gold standard to measure the cell's ability to get converted into cartilage, all right? Or it is a gold standard for chondrogenic differentiation. So they compared the gold standard with their approach. So here they measured the expression of SOC9 mRNA and normalized it to the expression of beta-actin mRNA. So I think so. This is beta-actin is the housekeeping gene. So they normalize the expression levels to norm generally expressed, like always expressing gene, that is beta-actin. It is a housekeeping gene. It is always expressed in all cells. Okay. So what they see is that in presence of TGF beta only, the cells are getting differentiated, you can see, because in printed constructs or in pellet culture where there was no TGF beta, there is no expression of SOX9. And also the expression of SOC9 is lower in printed construct in comparison to expression of SOX9 in pellet culture. Why it is so? It is because in pellet culture, the cells are very tightly packed. So the cell-cell communication is much more better, all right, through interaction of some cell-cell junction molecules. Like there is N-cadherin, N-cam. So these are the cell-cell junction molecules which can interact with each other better and communicate well through exchanging various molecules, right? So that is much that type of communication is optimal, like the best one for chondrogenesis to happen. Whereas in printed construct, the cells are bit far apart. The cells which are present inside the droplet, they can only do that. Whereas cells which are encapsulated in another droplet, all right, they are bit far apart from each other because there is a biomaterial shield, right? The agarose and all. So that is basically separating uh, many of the cells from each other. So that cell-cell communication is bit weak in printed construct, because of which you saw slightly lower expression of SOC9 mRNA in printed construct. So yeah, um, these are the key results of the paper that shows the power of the technique, the droplet-based technique, and that too they did in an oil bath, and why they did it. I hope that makes sense to you guys now. Um, you can also see they can print very high resolution structures by using very small droplets, like around 1 nanoliter. So that's a very good contribution to the field that people can use for patterning multiple type of cell at a very high resolution. So if you have any questions regarding the paper, now would be the good time to ask, because I'm done with my explanation of the paper. I'll upload the entire paper today, okay, along with all the lecture recording and notes like I always do, okay? And not today, maybe not today, maybe before Saturday, I'll also post an assignment, online assignment, which will contain questions based on today's lecture and as well as the last week's lecture. So the number of questions would be bit more. Like usually I give 10 questions, maybe 15 or 20 questions you can expect this time. So as we are covering two lectures, all right? So yeah. And also I'll give you a bit more time as well to complete that assignment. So don't worry. If you have any questions, let me know.
No questions, ma'am.
All right. Thanks for letting me know. Did you find the paper interesting? Did you find their work interesting? Do you think this work is interesting to you? Did I do a good job in choosing this paper? Any comments, suggestions, anything from your side? I just realized something. This is the first class that we're having on December. Yeah, okay, that's good. So, yeah, if there are no questions from your side, I'll then take my leave and see you guys next week. Meanwhile, do expect to find the assignment in your tabs and please submit it before the deadline. All right. Thank you for joining. I'll stop the recording and end the meeting. Meanwhile, you can also leave the meeting.